Hello everyone. In my previous video, I have discussed about the light phase of photosynthesis. In today's video, I'm going to discuss the light independent phase of photosynthesis. Okay, we can also call it phase 2. Okay, now light independent phase is also known as dark phase. Now, although it is called dark phase, it doesn't mean it occurs only in dark, but it simply means that in this phase, light is not required. Okay. But the products from light phase will be used in this phase. So what are the products that we got from light phase? That will be ATP and NADPH, right? So in this phase is also called biosynthetic phase. Okay, biosynthetic phase. Fine. Why this is called biosynthetic phase? Because in this phase, carbon dioxide fixation occurs by a series of enzyme catalyzed steps and glucose is formed. Okay, glucose is formed. What is the main purpose or main goal of photosynthesis? Is to make glucose. That is the food, right? That is the food. Now, these steps of reactions or series of reactions were first discovered by Melvin, Kelvin and Andy Benson. Okay. So, based on these two scientists, the biosynthetic phase is popularly known as Kelvin and Benson cycle. Okay. Now, as I have told you that light phase occurs in the thylakoid. The membrane, particularly in the membrane of thylakoid, right? Same way, the light independent phase occurs in stroma. Okay, it occurs where? It occurs in stroma. Fine. Okay. Now, we can divide Kelvin and Benson cycle into three steps. Okay. First will be carbon fixation. Second one is reduction. Okay. And the third one is regeneration. Regeneration. Now, these are the three steps we will get in Kelvin and Benson cycle. So before starting the cycle, we have to know a very important compound, that is ribulose bisphosphate. Okay, ribulose bisphosphate. Remember this compound's name. Okay, bisphosphate. The abbreviation is RuBP. Okay, RuBP. Okay. Carbon dioxide enters Kelvin-Benson cycle by this RuBP. Okay, ribulose bisphosphate. This is a carbon dioxide acceptor compound. Ribulose bisphosphate is what? It is a carbon dioxide acceptor compound. Now, for this, we also need a very important enzyme. Okay, that is ribulose bisphosphate carboxylase. In short, we are going to call it RuBisCO. very important. This enzyme is very important for plants. RuBisCO. Okay. Now, its full name, what I said, ribulose bisphosphate carboxylase. Now, RuBisCO can also show oxygenous property. Fine. We will remember this because this thing will help us in our future topics. Fine. Okay. So, we have divided Kelvin cycle into 3 parts. Okay. First, we can get the carbon fixation step. Okay, carbon fixation. In these steps, what happens? CO2 enters the Kelvin and Benson cycle. Okay, CO2 accepted, that is what? CO2 accepted is what? RuBP, right? RuBP with help of the enzyme RuBisCO. The enzyme is what? The enzyme is RuBisCO, right? RuBisCO. Here you can see carbon dioxide with the help of RuBP. Okay, and the enzyme RuBisCO. A 6-carbon compound is formed. Okay, a 6-carbon compound is formed. But the problem is this 6-carbon compound is very unstable. Okay, that's why this 6-carbon compound is broken down into 3-carbon compound. Okay, 3-carbon compound, that is PGA. Okay, PGA. PGA's full form is phosphoglyceric acid. Okay, phosphoglyceric acid. Now, in this cycle, the first stable compound is a 3-carbon compound. That's why it is also called C3 cycle. Okay, that's why it is also called C3 cycle. We will also get C4 cycle, CAM cycle, etc. But this is C3 cycle. Why? Because the first stable compound is PGA, 3-PGA, that is a 3-carbon compound. Okay, after this what happens? Reduction takes place. Our second step, that is reduction. Okay, reduction. In reduction, what happens? 3-PGA forms G3P. Okay, that is glyceraldehyde 3-phosphate. Okay, that is glyceraldehyde 3-phosphate. Now, as we can see here, 3-PGA is converted into G3P, that is glyceraldehyde 3-phosphate, using ATP and NADPH. Okay, ATP and NADPH. Fine. And glyceraldehyde 3-phosphate is made. Okay. Now, these glyceraldehyde 3-phosphate, some of this glyceraldehyde 3-phosphate will leave the cycle to form glucose. Okay. Some G3P will leave the cycle to form glucose, and 5 molecules of G3P will form RuBP. Okay. Now this RuBP will continue the cycle. This RuBP will continue the cycle. So in reduction phase, what we are getting, that 3-PGA is forming G3P, glyceraldehyde 3-phosphate. Okay. And what is used here? ATP and NADPH. Okay. Okay. And some G3P is used to make glucose. Okay. And the remaining G3P is used in the next step, that is regeneration, and G3P regenerate RuBP. Okay, G3P regenerate RuBP, and again the cycle continues. So this is a very simple explanation of Kelvin cycle, and you can get the flowchart from here. And yes, the overall reaction is given here, that is 6CO2 plus 12 NADPH, 18 molecule of ATP is used here, glucose is produced, NADP plus is produced here. Now this NADP plus will be used in light reaction again. Fine, we have seen that NADP plus is used in light reaction to form what? To form NADPH, right? Again you can see here ADP is used, again it will be used in light reaction. So this is a continuous cycle. Fine. This was a very simple explanation of Kelvin cycle. That's all for today's video. In next video we are going to start a new topic. If you like my content, please like, share and subscribe. Thank you.